Hello, everybody. How are you all? Welcome to our lightning talk presentations on the ecology and conservation of primates. My name is Emilio Bruna. I'm a professor at the University of Florida and president of the ATBC. I hope you're all having a great time at our virtual meeting. We have an outstanding lineup of speakers today. I'm really excited about this. This is really an opportunity for me to learn a lot about a subject I'm interested in from some real experts. I am going to go through some logistics here very quickly. I'd like to introduce our speakers in this video format. It's a little difficult to give them the recognition sometimes that I feel they deserve. So we're going to introduce them all, and then I'm going to give everybody just a little bit of information about how to ask questions of the speakers during the presentation, if that's okay. So again, for the lightning talks on the ecology and conservation of primates, today's speakers are Veronarindra Ramanajatto from the Université d'Antananarivo in Madagascar, Simisento Hasimija Mihaminekena from the Aspinall Foundation, also in Madagascar, and Sophie Dalmeida Silva from the Primatology Lab at in Rio Claro, it's in Brazil, at Unesp. Diogratias Tuyesingizie from the Dian Fossey Gorilla Fund, followed by Robin Morrison, also from the Dian Fossey Gorilla Fund. Felipe Buffalo from the Primatology Lab at Unesp. You guys might want to take a break here. There's going to be a couple of speakers here coming from Unesp. We have Felipe Buffalo, also Eduardo Zanetti. Gabriela Rezende will come after that, followed by Jacqueline Ballantyne from the University of Miami, Eric Ndayishimiye from the Dian Fossey Gorilla Fund in Rwanda, and wrapping it up for us today is Deborah Giancola from Unesp São Paulo, the Institute of Biosciences. Thank you to all of you for joining us today. This is really exciting. If you have any questions at all for the speakers, please post them using the Q&A button on your Zoom screen, not the Whova function for now. We won't see them if you're posting them on the Q&A section on the Whova for the lightning talks. If we don't have an opportunity to get to your question, or you'd like to post questions later for your speakers, you can do that via Whova. But for now, please do post your questions via the Zoom Q&A button. And with that, thank you all again. And I'm going ahead and mute out here, and we'll get started with the presentations.
Hi everyone, my name is Fer and I'm from Madagascar. Today I'm going to talk about the structure of microhabitats used by the rufous mouse lemur across a heterogeneous landscape. Structural variation in microhabitats, such as differences in vegetation composition, forest layers, and plant growth forms, can influence primate abundance and diversity in a forest. Besides, the preference for specific microhabitat structures among different primate species can maintain the coexistence of diverse primate species in a given area. However, the current vegetation alteration and recovery have built heterogeneous forest landscapes of degraded, recovered, and intact habitats, which may influence microhabitat preference in primates. We thus conducted microhabitat analysis to investigate the preference of the rufous mouse lemur in Ranmafana National Park, during which we compared microhabitats where we captured them to where we did not. The first question we wanted to answer is, what aspects of microhabitat structure do the rufous mouse lemur use across a heterogeneous forest landscape? We compared the diversity, the size of trees and shrubs, and the presence of lianas in 132 used microhabitats with that of 240 unused microhabitats. In less disturbed forests, we found no significant differences in both diversity and structure. However, in both habitats, we found that used microhabitats have higher plant diversity and densities of trees and shrubs of large diameter. In disturbed forests, used microhabitats differ from unused in three structural metrics. Specifically, secondary forests used microhabitats presented significantly lower cover of the herbaceous layer, whereas in forest fragments, used microhabitats have a higher density of medium diameter plants, but lower densities of understory trees and shrubs. The second question we sought to answer is, how does the structure of the microhabitats used by the rufous mouse lemur vary across forest types? We compared the diversity and structure of only used microhabitats of each forest type to that of the primary forest, assuming that the relatively intact state of the primary forest is an ideal habitat. First, we found that compared to the microhabitats in the primary forest, plant diversity decreased in all other forest types, but it was only significant for secondary forest and forest fragments. Second, diameter decreased in all habitats. Specifically, it was the density of plants with DBH of 5 to 10 centimeters that significantly decreased, which is then compensated by an increase of the densities of extra small and small-sized plants. However, plant height increased, except in regenerated forests. In disturbed forest, we noticed a significantly increased density of tall trees and the cover of the herbaceous layer. Finally, trees and shrubs with liana significantly decreased compared to the primary forest, except in secondary forests. Specifically, we found a significantly lower densities of trees and shrubs with one liana in other forest types. In short, the rufous mouse lemur used small and medium-sized plants without lianas in primary and regenerated forests, while changed to extra small and small-sized plants with one liana in secondary forests and fragments. Although this study focuses on one primate species, it has shown how important the examination of potential changes in microhabitat can be. Such results could be used in conservation to predict where species could occur in a heterogeneous landscape, but also could optimize the long-term preservation and management of threatened species. And that closes my presentation. Thanks for watching. If you have any questions, please feel free to contact me.
Hello everyone. I am Hasimija. I come from Madagascar. I'm here to talk about the home range study of greater bamboo lemur, a comparison between the wet and the dry season. First of all, of kinds of mammals is the greater bamboo lemur. It is a lemur species which rely especially on bamboo for diets. It is an endangered species due to its unique diet. So, the greater bamboo lemur is a lemur bamboo specialist. His distribution is very restricted in south and southeast of Madagascar, and it is an endangered species. We can also find the greater bamboo lemur in Sahavola site, which is located in the east of Madagascar, and it is a highly disturbed habitat around 30 hectares and a private property. To monitor the two groups of greater bamboo lemur in Sahavola, we have monitored. We used the Atman sampling every 5 minutes, and to delimit the home range, we have used the minimum convex polygon. As a result, we have found that during the dry season, the size of the home range of the two groups tend to decrease in comparison to the wet season. But this difference is not significant. In contrast, the size of the two groups is decreasing to the dry season, and the difference is significant. So we recommend to develop a strategy to monitor and protect population of glitter bamboo lemur, and the use of other methods to delimit the home range of these groups of lemurs in large scale, for example during 1 year. As a conclusion, the glitter bamboo lemur is an endangered species which rely on bamboo for diets. Its distribution is very restricted in south and southeast of Madagascar, and the size of the home range tend to decrease during the dry season in comparison to the wet season. So we recommend to ensure the conservation of the glitter bamboo lemur for developing strategy to monitor and the use of other methods to limit their home range. That's all for me. Thank you for your attention.
Hi, I am Onja, and our study is entitled The Role of an Endangered Small-Bodied Primate Species with a Sea Disperser in the Atlantic Rainforest. The Brazilian Atlantic Forest is a global biodiversity hotspot with high beta diversity and multiple plant-animal interactions. However, most Atlantic forest areas show high levels of defaunation. The selective and accelerated removal of larger frugivores has changed local patterns of communities, which are currently composed mostly by small species. Although small species are not able to swallow larger seeds, larger dispersers also ingest medium and small seeds. And when larger dispersers are absent, small frugivores become the only dispersers of these seeds. Some of these small frugivores are also endangered, and their importance on seed dispersal networks are largely unknown. Here, we evaluated the role of an endangered small-bodied primate species within the seed dispersal network of its distribution area. The black lion Tamarin BLT is a frugivorous faunivorous species endemic to the São Paulo Atlantic Forest. Despite being an endangered species, it persists in intensely fragmented areas. Also, by using large worm ranges, traveling long daily distances, and consuming a great diversity of fruit species, makes it a potential seed disperser. And how did we do it? We determined the list of plant species the BLT disperses by monitoring three groups in 2 areas between 2018 and 2019. Then we combined the data collected on the field with all studies available. We extract frugivore data of the other animal species from the data paper of Bello and collaborators. This resulted in a matrix with 570 on potential interactions. From that, we evaluate the network using 6 parameters, structural parameters. The network showed medium levels of nestedness, modularity, robustness, and low level of connectance, showing an asymmetric system of interactions was observed for most mutualistic networks. 23% of plant species showed maximum dependence on the interaction with BLT, especially from Bromeliaceae and Cactaceae families. The network showed 7 models, and the capuchin monkey, Sapajus nigritus, the white roller monkey, Aluatogorba climitans, and the Jacupemba, Penelopes superciliaris, were the species most functionally similar to BLT, occupying the same model. Subgroups of dispersers linked to subsets of species in its model could compensate in parts the functional loss of another disperser within the model by performing similar ecological roles. However, literature data show that these compensations are limited. These species plus the tapir, Tapirus terrestris, were the species that most contributed to network maintenance. Despite the size difference, BLT was able to disperse medium seeds common to megafauna species. We know that as the size of the seeds increase, the potential dispersers decrease. Our results suggest that BLT can exert an important role as disperser, mainly by sharing dispersal of medium-sized seeds with larger frugivores, which may be extinct in most fragments of the Atlantic forest. Thank you.
Hi everyone, my name is Deogratias Twisingize. I work for the Dan Fosse Gorilla Fund. Today, I would like to talk about food availability influences bathing seasonality in endangered golden monkeys. The endangered golden monkey is a subspecies of blue monkeys which are found in Africa. This subspecies is restricted in 2 different fragments, including the Virunga Massif, which is located in Rwanda, Democratic Republic of the Congo, and Uganda. The other habitat of the golden monkey is the Gishwati forest in Rwanda. Here, we have followed 3 different groups, including Group K and M, located in the Virunga Massif, and Group G, located in the Gishwati forest. In the Virunga Massif, golden monkeys are mainly found in the bamboo zone, while in the Gishwati forest, golden monkeys are mainly found in the Afro-montane forest. And little is known about how these 2 populations adapted diet and reproduction in these 2 different fragments. We have collected demographic data specifically on birth events between 2004 and 2018 in the groups located in the Virunga Massif. In the Gishwati forest, we have collected the birth events data between 2017 and 2018 in the Group G. We also collected the feeding data between 2017 and 2018 in all the studied groups. We collected the phenology data on key food plant eaten by golden monkeys between 2017 and 2018. Where in the Virunga Massif, we have collected data on the bamboo shoots regeneration, while in the Gishwati forest, we have collected the data on fruit trees. As the results, groups located in the Virunga Massif have adopted a frivolous diet, and the bamboo shoots are their preferred food plant item. In contrast, the Gishwati group has adopted a frugivorous diet supplemented by leaves and other food plant items. Key food were eaten seasonally once they were available, with bamboo shoots being eaten by the golden monkeys of the Virunga Massif and fruits being eaten by the golden monkey located in the Gishwati forest. Key foods determined birthing seasonality in golden monkeys, where group M gave birth mainly between February and March, and group A gave birth mainly between September and November, while group G gave birth between March and May. Our results shown that food availability shapes production in golden monkeys, where females golden monkeys give birth prior and during bamboo shoots and fruits availability. Therefore, the timing of birthing reflects variation in availability and consumption of key food. What is interesting is that groups located in the Virunga Massif have two different birthing seasons. These reflect variation in key food plant species such as bamboo shoots located in the Virunga Massif. Our findings show the importance of bamboo shoots and fruits in the reproduction of golden monkeys. However, there are detectable changes in key food regeneration, which is a possible effect of climate change. Therefore, we would like to recommend continuing monitoring in order to more understand the effect of changes in key food availability on long-term survival of the golden monkeys. Thank you so much.
Hi, my name is Robin Morrison, and I'm a postdoc with the Dian Fossey Gorilla Fund, and I'm going to talk to you today about respiratory disease transmission in mountain gorillas. So a lot of research in the last 20 years has demonstrated the terrible effects that infectious disease is having in wild ape populations. And partly this is due to the fact that they're so closely related to humans and can catch many of the same diseases as us. And this is likely to be exacerbated further by COVID-19, which we know that gorillas can also catch. So we need to find ways to reduce this problem. And one strategy is to use data from past outbreaks, using things like social data and health data, to understand how diseases are transmitting through these wild populations, and using this to guide our conservation strategies. I work at the Karasohi Research Center in Rwanda, where scientists have been monitoring mountain gorillas since 1967. And this means there's a huge amount of data on things like their health and also the patterns of social interaction, which is just the sorts of data we need to understand how diseases are transmitting in the population. So since 2004, there have been 15 respiratory infection outbreaks in the population that we monitor. And we also have a huge amount of data on the patterns of social interaction during that same period. So we collect data on which individuals are in physical contact and also how often individuals are within 5 meters of each other. And so we can use all of this to build social networks to understand the patterns of interaction going on in the population when these outbreaks happen. So for each outbreak, we built 2 social networks, one based on proximity and one based on contact. And we looked at whether we could predict who would get sick during these outbreaks based on the social network. First, we looked at whether those with symptoms were more closely connected in these networks than we would expect by chance, but we didn't find that this was the case. Then we also looked at whether those with higher network centrality, so those that were better connected in their groups, were more likely to show symptoms. But again, we didn't see this. And this is really different to what we see in most other animal species and most other outbreaks. In a number of these outbreaks, we were seeing that the diseases were just transmitting incredibly quickly. So, for example, almost all of this group becomes infected by only day 3, showing that this network of really interconnected individuals with lots of social interactions enables these diseases to spread really quickly. In other cases, they spread a little bit more slowly, and to begin with, they tended to spread to the social contacts of those that were already infected, but it rapidly spread throughout the group. We also looked at the capacity for disease to transmit between different groups. And we did this looking at transfers, when a gorilla moves from one group to another, encounters where these groups approach each other, or range overlaps. So to what extent the area used by one group overlaps with the area used by another group. What we found is that Transfers were taking place in 20% of outbreaks and encounters in 27% of outbreaks. So these were fairly common, but in none of these instances did they cause an outbreak in a previously uninfected group. When we looked at range overlap, we found no evidence that simultaneously infected groups had greater range overlap with each other than those of uninfected groups. And so this is really not what we would expect at all if gorillas were infecting their neighbors. So we haven't found any evidence between group transmission of these respiratory infections. So how are they reaching new groups? Well, genetic evidence has demonstrated that in 2 of these outbreaks, they've actually been caused by pathogens that are human in origin. So perhaps the most likely source of each of these 15 outbreaks might actually be independent introductions from humans. So in conclusion, what we're finding is that there's this really rapid and unpredictable transmission going on within these groups, and they're highly interconnected in the social network. So it's really difficult for us to limit this transmission, because if we cut off one transmission pathway, there'd be so many other alternative routes. But what we do find is that between groups, there's no evidence of transmission going on. So their social structure in that regard seems to be protecting them to some extent. So instead, what it seems to show is that actually it's humans that are responsible for these repeated introductions, and that if we want to best protect this population, what we need to do is make sure we are incredibly careful during human-gorilla interactions. So things like wearing masks, keeping a serious distance when possible, and also ensuring everyone is vaccinated and tested if they can be. Thank you very much for listening. And I'm really happy to answer any of your questions. I need to say a very big thank you to my co-authors on this project, Yvonne Mushimiyamala, Taras Duinsky, and Winnie Eckhart, and a huge thank you to everyone at the Diane Fossey Gorilla Fund who's helped collect this data and make this all possible. And also a thank you to my team in Exeter as well. Thanks for listening.
Hello, my name is Felipe Buffalo and my presentation is entitled Route Planning Process by the Endangered Black Lion Tamarin is influenced by resource distribution and spatial limitations. On a daily basis, besides displaying complex social behaviors, primates execute a wide variety of decisions concerning territory defense, predation avoidance, and where and when to feed. In turn, these decisions affect directly the daily routes primates execute through their home ranges. However, our knowledge of what are the main factors influencing these decision processes is still limited, above all in forest fragments. Among the primate species in the Atlantic forest, the black lion tamarin is only found in the state of São Paulo, restricted to small fragments. It is a small primate species considered threatened by the IUCN Red List of Threatened Species. It is highly frugivorous, highly territorial, and known to travel long distances daily. Our aim in this study was to understand what are the main factors influencing black lion tamarin's route planning process in a 100 hectares forest fragment. Due to its highly frugivorous diet, we expect daily routes to be determined by fruit feeding sites. Also, since the study area consists in a fragment, we also expect routes to be restricted by the physical limits in the area. We followed a fully habituated group for 42 entire days, from dawn to dusk. We recorded every 5 minutes the group's location as well as the group's behavior through scan samplings. We identified and recorded the location and duration of all fruit feeding events as well as intergroup aggression events. To understand how the group used the area, we divided the home range into 3 zones. With 2 100-meter buffers, we created a fragment edge zone, a territory border zone, and between them, a central area. We used the Kruskal-Wallis test to compare the use between the zones. To assess the distribution of fruit sites, we estimated empty space distances between random locations and the nearest fruit feeding site throughout the home range during the study period. In order to identify the locations responsible to orient the routes, we use the change point test, developed by Burning colleagues, which allows an objective assessment of significant locations for change of direction in daily routes. These could be landmarks, travel goals, or physical barriers. The group used an area of 46.8 hectares and traveled daily on average 1,700 meters. Frugivory was the most recorded behavior, corresponding to almost 30% of our scans. However, it was not as much associated to change points, being locomotion and foraging the behaviors mostly associated to the locations of direction change. We recorded one event of intergroup aggression every 4 days, which is a frequency almost 3 times higher than the observed in a continuous forest area. We found a mean empty space distance of 33 meters throughout the study period, suggesting that if we consider the mean step length of 20 meters recorded, the group would be able to reach a new fruit site when traveling less than 10 minutes in any direction in the home range. Although it was not significant, we found a higher proportion of change points, here represented by the colored shapes, in the border zones, indicating a possible influence of the physical limits of the area and territory defense on route planning. Our results indicate that, despite the high importance of fruits for the group's diet, fruits did not represent an important factor for route planning process, probably due to its dense distribution throughout the area. However, we registered long distances traveled daily, corroborating the importance of this behavior for territory management and resource monitoring. Change points were mostly associated to traveling and foraging, with a higher proportion in the borders of the home range, suggesting a high importance of the social behaviors and physical limits of the area for the route planning process of black lion tamarins in this area. In conclusion, while previous studies in larger areas usually point fixed feeding sites as the main factor shaping primates' trajectories, we suggest that route planning process might be context dependent. I would like to thank for all the support I had during this research and also for all the attention during this presentation. I will be glad to answer any questions.
Hello everyone, my name is Eduardo Zanetti and I am a master's student at Unesp Rio Claro, Brazil. Today I will be presenting some research we have been conducting recently and which is entitled Black Lion Tamarins Movement Patterns in Distinct Environmental Contexts. We know that fragmentation in habitat loss reduced continuous forest patches to smaller and diversely shaped fragments. But what do we know about the effect of patch size and shape on primate movement patterns? Well, not much. Patch size reduction usually enhances the percentage of time spent feeding, but its effect on the other behaviors such as resting and movement patterns are inconsistent. Furthermore, some studies show that ranging patterns become much more circular as the patch size gets smaller. This results in a presumed, but not often assessed, effect on ecosystem services such as seed dispersal. In this research, I'm interested in exploring what black lion tamarins can tell us about this question. BLT, or the Black lion tamarin, is an endangered primate according to the IUCN. It is frugivorous and insectivorous, and its daily travel distance reaches a mean of 3.5 kilometers. Their home ranges varies from 30 to 400 hectares, and their high degree of frugivory is associated with the effective dispersal of seeds. They are also a highly territorial species. Also, we know that BLT home ranges correlate strongly with fragmented size. Then our question of research is, how do different patch sizes and shapes affect the BLT movement patterns? With the aims of comparing movement patterns in 4 areas, we hypothesize that smaller and riparian forest fragments restrict BLT movement patterns. Our predictions are that those special restrictions lead to a smaller step lengths in comparison to large forest patches, to less directional movements in smaller patches but still some directionality in riparian forests because they are usually very narrow, and to larger step lengths and directionality when heading to sleeping sites. We collected data on 4 areas throughout the BLT geographical distribution in São Paulo State, southeastern Brazil, and classified them according to patch size: 1 small, 1 medium, and 1 continuous forest, and 1 relatively small riparian forest. We collected data in varied years and months and corrected GPS errors in the trajectories. We used complete from sleeping site to sleeping site and incomplete days. When relocations were lost, a new trajectory was defined, and we used the Ad Habitat LT and the Tidyverse collection of packages in R. Here are some BLT complete daily routes. Please bear in mind that these plots are not in scale. Step lengths were larger in continuous forests and smaller in riparian forests, and were similar in small and medium patches, although all areas differed significantly. Relative angles were not uniform. Thus, all areas present some directionality. Relative angles differed between all four areas, being much more directed in continuous forest, followed by the medium-sized patch, then riparian, and then the small patch. This last one, which showed the more random directions of traveling. Furthermore, we also selected some specific behaviors. As we were interested in directionality in frugivory, we selected 3 steps before feeding. For this behavior, movement patterns follow the general pattern. It is step length was larger in continuous and smaller in riparian, being similar in small and medium-sized patches. And directionality was lower in the small patch and high in the continuous forest. We saw the same pattern for foraging behavior, both in terms of step length and relative angles. And for 6 steps before arriving at the sleeping site, we showed that BLTs are faster when traveling to sleeping sites, a speed of around 25 meters traveled each 5 minutes, indicating that BLTs cover a lot of space in the last moments of the day, especially in continuous forest. Our study suggests that ranging pattern seems to be more dependent on fragment size and shape than on behavior. It also suggests that continuous patches with low BLT density results in larger home ranges, forcing them to travel faster throughout their home ranges in order to defend it. Furthermore, riparian forests make movement patterns lower but more directional when compared to small fragments similar in size. And for last, we saw larger step lengths when heading to sleeping sites. Our predictions were correct for the general patterns of movement and for most of the specific behaviors. Thus, we suggest that fragmentation and habitat loss alters BLT ranging patterns with possible effect on their ecosystem services, for example, diminishing sea dispersal distance. Some recent studies show a direct effect of resource distribution on ranging patterns, and this will be the next step of our research. I'd like to thank my lab team and all field assistants that collected those data. I'd like to thank the ATBC Congress organizer and the agencies that funded my research. Please contact for further questions and collaborations. Thank you and be safe.
Hi, I'm Gabriela Rezende, a PhD candidate at the São Paulo State University in Brazil. I'll be presenting my work entitled Black Lion Tamarins Do Not Change Daily Distance in Small Forest Fragments, but Reduce and Share Their Home Ranges. The movements and space use of forest-dependent species are fundamentally driven by forest structure, as this is a proxy for resource availability. The movement capacity of an animal determines its efficiency in resource gathering and thus its survival under changes in the environmental circumstances. So quantifying the patterns and drivers of species movement in degraded forests due to fragmentation can inform guidelines for better habitat management and increase population viability. The black lion tamarin is an endangered primate endemic to the Brazilian Atlantic Forest of São Paulo State. Its biggest threat is fragmentation and lack of connectivity among forest patches. Given it relies on trees for moving and surviving, understanding the influence of forest structure on its movement and use of available habitat can provide invaluable information to support its conservation. In this work, we aim to characterize and compare the space use and movement patterns of black lion tamarin groups living in forest patches with contrasting forest structure. We monitored 4 groups using GPS devices deployed on 1 tamarin per group. There were 2 groups from a large fragment called Ponte Branca and 2 from a small one called Guarei. Group movements were monitored over a month with GPS locations recorded every 10 minutes during their activity period. We collected an average of 1,152 locations per group, over 10 to 22 monitoring days. To account for the differences in vegetation structure between the areas, we characterized the vegetation from each forest patch by measuring height and diameter at breast height of all trees with DBH larger than 4.8 centimeters in 22 plots per study site. From these measurements, we estimated 6 variables that we used to test the difference between the areas. All estimated variables were higher in the smaller fragment. From the locations recorded, we estimated home range size for each group using 95 fixed kernel utilization distribution for total home range and 50% for the core areas. We found that in the small fragment, the groups presented a smaller home range and core areas, with up to 37% overlap between the groups, including the core areas, which was not the case for the large fragment. We also estimated mean daily path and found that, despite the up to 5-fold difference in home range sizes, the daily distances were similar between the groups, averaging about 2.6 kilometers per day. Finally, we compared the distance of the locations from the forest edge among the groups and also the same distance against random locations. We found significant difference between the 4 groups. And when compared against random locations, we found that in the small patch that is shown on the top, the group 1 remained farther to the edge than would be expected by chance, while group 2 remained closer. However, in the large fragment, shown on the bottom, distance from the border did not differ from random expectation in both groups. In conclusion, the estimated home ranges for the groups in the small fragment are among the smallest ever recorded for the species, with also a high overlapping between neighboring groups. This suggests a strong effect of patch size on home ranges. An important next step now is to quantifying the relative effects of competition, edge effect, and resource availability driving these differences between forest patches. I would like to thank all the institutions that has been supporting this project, and also you for your attention. I'll be happy to answer any questions you might have. Thank you.
Hi, my name is Jacqueline Ballantyne, and I'm a PhD student in the Defaunation Lab at the University of Miami. The project I will be presenting today is entitled Infested Fruit Consumption in a Vertebrate Frugivore, the Golden-Backed Wakari Monkey. I was focused on the feeding ecology of frugivores. I wanted to look at nutrient balancing, insects as a food source, and covert carnivory. Insects make up a significant part of the diet of a wide variety of primate species. However, ingestion of insects by primates through consumption of infested fruits is considered widely underestimated. As larvae are commonly 60 to 80% protein, infested fruit selection may be an important means for primates to access protein not otherwise easily available from fruits and seeds. We consider this topic important for understanding primate foraging decisions and suggest diet sampling methodologies that should be considered for this foraging mode. Methods for detecting seed predation could be employed both within and outside primatology to recalibrate the contribution of fruit-inhabiting insects to diets of species considered predominantly frugivorous. My major research questions were what predicts the selection of infested fruits in sea wakari, and how do they find and identify infested fruits in their environment? Specifically, I looked at tree-specific traits and other factors that may have contributed to these questions, and fruit-specific traits such as volume, mass, and longest length. The methods we used for this research involved fruit measurement and observation of those ingested by sea wakari, IVLA values of their selection, preference, or avoidance, and we used general linear mixed effects models in order to analyze our data. So the field site, the data was collected by Dr. Adrian Barnett, the vast majority of the data, and colleagues between October 2006 and July 2008 in Jau National Park, northwest of Manaus. So some of the results we found were that in some cases, sea wakari does prefer infested fruits over non-infested fruits. So of these species that we looked at, a few that had avoidance were Amanoa oblongiflora and Mabia nitida. And then there was obviously a lot more preference for infested fruits that we observed in these species. However, this data set is quite biased in what we were able to observe over the study period and may not reflect the actual proportion of infested fruit selection in this species. To understand which factors influence the foraging decisions of sea wakari, we looked at the nested decisions between tree-specific factors and fruit-specific factors. When looking at what factors may predict the selection of infested fruits in Siwakari, we did not find any significant correlates within our tree-specific factors. However, when we looked more generally at the factors that may predict infestation status in these fruits, we did find strong predictors of infestation status, including canopy size, the proportion of trees infested per hectare, and the phenophase consumed. And then we also asked, how do wakaris find and identify infested fruits in their environment in terms of their fruit-specific factors? And we saw that length, mass, and volume were all significantly related with infested fruit status in the fruits selected by wakaris. As you can see in these graphs here, with length increasing, infestation status also increased, with mass increasing, infestation status decreased, and with volume increasing, infestation status increased. This study had many limitations, as it was a preliminary look at infested fruit consumption in Siwakari and part of a much larger study to look at their diet in general. Future research may focus on infested fruit consumption with experimental trials and specific measures of selection not encompassed in this study. I thank you so much for joining me today, and I will see you soon.
My name is Eric Ndaishime, a research assistant at the Dana Fosse Gorilla Fund. This study investigated crop raiding conflict in golden monkeys of the Volcanoes National Park in Rwanda. Crop raiding is one of the main causes of human-wildlife conflict, challenging conservationists worldwide, and primates are frequent raiders around many protected areas. Crop raiding negatively affects not only farmers' livelihoods, but also the conservation of the involved primate species. So why golden monkeys? They are endangered. They remain only in two isolated populations, one population in Gishwati Mukura National Park and the other one in the Virunga Massif. This study focused only on golden monkeys of the Volcanoes National Park, which is part of the Virunga Massif, as you can see on this map. As you can see on this photo, the Volcanoes National Park edges are directly adjacent to farmlands and a dense and poor human population. The golden monkeys in the Volcanoes National Park basically inhabit the bamboo zone, which is the nearest to the park boundary. No records of crop raiding by the monkeys exist until at least the mid-90s. But recent studies list golden monkeys as second raiders, and the frequency by which they leave the park to raid on potatoes is increasing over time. Moreover, golden monkeys challenge the existing mitigation measures. Integrated approach are recommended to attain a good understanding of human-wildlife conflict scenarios and to plan for alternative mitigation interventions. We used an integrated approach to investigate to which extent golden monkeys crop raiding affect their conservation and farmers' livelihood, and to seek for possible alternative mitigation measures. We used a structured survey, workshop, behavior observation, and observation experiment. The key survey outcomes were that 95% of participants had experienced potato loss to golden monkeys. 36% had used potentially harmful methods to stop raiding monkeys, like throwing stones to raiding monkeys. And a few cases of adverse impact on monkeys were described, like some monkeys were injured while being chased by farmers. Of the 5 measures that were discussed during the workshop, crop guarding was perceived as the most effective mitigation measure. But the existing crop guarding needs to be improved through a government financial support, and properly organized guarding in cooperative is also needed. Monkeys were less social, they vocalized less, they self-groomed less, the infants suckled less, They were more vigilant when on farmland compared to when they were inside the park, reflecting a high level of perceived risk by the monkeys when outside of the park. In the test aversion experiment we tried, we used chili oil and potatoes. The potatoes smeared with chili oil were put outside the park as shown by this experimental area design you can see here. The monkeys did not eat potatoes smeared with chili oil. They fed on unattained leftover potatoes instead. This suggesting that the smell chili could potentially deter the monkeys from eating crops. In conclusion, the survey outcomes and the behavior changes shown by the monkeys on farmland indicate that crop raiding by the monkeys can not only affect negatively a farmer's livelihood, but also the monkey conservation. Overall, our approach showed that the existing mitigation measures like guarding need to be improved, while alternative measures like test aversion should be further explored. This will facilitate a positive coexistence between the monkeys and farmers and will ultimately improve their conservation. Thank you for your kind attention. I extend my thanks to my co-authors, to the institutions and organizations that supported this study. If you have questions, you are very welcome.
Hi, I'm Débora Tomiati Oncola, biologist from the São Paulo State University in Brazil, and I will present my work, which is called Effect of the Habitat Quality on the Sleeping Site Selection by the Black Lion Tamarin, Lantoptex chrysopicus. So neotropical primates are usually arboreal, using trees as sleeping sites too. They can be in the shape of hollow trees and tangle of lianas. Their choice is influenced by characteristics such as competition, predators, parasites, proximity, and availability of food sources. Habitat quality in this study is the ability of the habitat to provide resources for the survival of individuals such as food, shelter, and protection. Primate populations in tropical forests are threatened nowadays by the loss of habitat to the destruction of forest and its fragmentation. So how the process of resource selection varies within the habitat quality allows us to have an idea of the ecological plasticity of the species. So we analyzed this process in population of black lion tamarins, an endemic endangered primate of the state of São Paulo in Brazil, that are found in a highly untrusted landscape in Atlantic Forest. Today, those areas hold around 1,600 individuals. In this study, we tested whether and how the sleeping site selection process was altered by habitat quality, comparing groups of continuous and fragmented forest areas. This knowledge allows us to trace strategies to contribute to the conservation of its populations, such as plantation of preferred tree species as sleeping sites, identify suitable areas for translocation and reintroduction, and to foster decision-making for conservation projects, mainly in Atlantic Forest. So we compared two groups, one in a continuous and another in a fragmented forest, as can be seen here in a map. The data collection happened during research carried out at the Laboratory of Primatology. The sleeping site availability was provided through the implementation of 20 plots in each area. Some of the data collected were species, total height, diameter at breast height, DBH, and the number of connections with other canopies. At the sleeping site were also collected the type of den and the height of the entrance. Comparing the sleeping site trees with those available in the environment, the groups chose sleeping sites with familiar characteristics, such as trees taller than those available and with fewer connections between the canopies, evidencing the selection of characteristics that provide protection against predators. Comparing the selection between areas, DBH in the continuous forest is greater than the DBH average of the fragment. The height of the entrance was also greater in the continuous forest, as can be seen here. Simpson's analysis showed no significant difference between the species diversity of the areas, and looking for the effect of resource availability, we found no difference between the forest structures. Then they presented little distinction between the raised characteristics. Thereby, we can suppose that the size of the fragment does not necessarily inform the availability of resources, but is influenced by the level of degradation within the area. Looking for the sleeping site selection analysis, in the continuous forest, the primate mostly used hollows, while in the fragment, lianas. So the lower number of tree hollows in the fragment, usually considered preferable by the species, indicates a possible paucity of this resource, with a known consequence to the populations yet. The preference of hollows can be explained by its safety, providing protection against predation. And to conclude, the forest structure was similar between the two areas, and the fragment was capable of supporting the populations without significantly impacting the sleeping site selection process. Meanwhile, it is important to point out that fragmentation impacts the species in many ways, and studies are necessary to assess those impacts on the ecology of black-lion tamarin's populations, such as locomotion, dispersion, and behavior. And now I would like to thank those institutions and everyone who helped with this project. Also, thank you all for the invitation, and I'm available to answer any questions.
Well, we're back, and I want to thank all our presenters. That was really fantastic. I learned a lot. I have a bunch of questions here, some of which came to me directly. We have a couple that we posted to each other as speakers. I'd like to, the first one is actually from this guy right here, Gustavo. You want to come in? So this is Gustavo Cauê, and he loved your presentations. He had a fantastic time, and he says he's considering a career in primatology. And his question is actually for Felipe. Felipe, Gustavo wants to know what was the cause of the intergroup aggression, and his idea is that maybe it's that you have trees with a lot of fruit that they're fighting over, given that they spend so much of their time eating fruit. He thought that might be the cause.
Thanks for the question. Yeah, there are many factors that could affect and influence intergroup competition and aggressions during the daily pets, But we had many during our study, and many were actually related to feeding events. So one group wanted to go to a feeding tree, possibly important one, and met another group feeding there, and they would fight for the resources. And also black lion tamarins are highly territorial, so they usually mark the territory and vocalize a lot, and many times when one group is vocalizing at a specific location on their home range, another group listens to that and comes to fight for the territory, and try to demonstrate that they are indeed living there and trying to manage the space on a restricted area.
Fantastic, thank you. Got it? Got it. All right. Well, thank you. Then the next question here is for Dio Gracias. And actually, this one was something the two of us were talking about during that presentation as well. Group G's diet was so different. And we were reading the graphs at the first two pie charts and comparing them. Why was it that the third group's diet composition was so different as opposed to the first two, which were, it seemed like, almost over 95% bamboo shoots?
Yeah, thank you so much. The group K and M live in the Virunga Massif, which is mainly dominated with non-fruiting trees, while the group G living in the Gishwati forest, the Gishwati forest is dominated with the fruiting trees. So that's why the diet is so different.
Fantastic. Thank you very much. We have a question here. Just a moment. I'm going through it. Okay, so would it be possible to, here we go, finally. For Robin Morrison, Robin, I'm trying, yes, there you are. Robin, could you have you considered the possibility, the question is, that groups are getting infected by saliva that's found on leaves, fruits, and branches?
Yes, yeah. So we sort of looked into this by looking at home range overlap. So it was one of our kind of one of the hypotheses we expected probably less would be harder to transmit, right? It's harder to kind of eat the same fruit as someone else and then get sick from it in relation to kind of just walking past another gorilla or another human. But we did look at it in terms of home range overlap, right? So if the group's home ranges overlap more, there's a higher likelihood that an individual from one group will eat something and leave it behind, and another individual from another group will come along and eat it. And we just found no evidence of that at all. We found like actually the direction was the opposite. It wasn't significant, but there was less home range overlap between concurrently infected groups than groups that weren't infected, which was pretty weird. So we took that as fairly strong evidence that this wasn't going on, but it is a genuine possibility in kind of other areas.
Would you mind if, since we have you here, if I asked a follow-up question here, a related question? I was, I think probably like a lot of other people, really surprised to see that range overlap wasn't that significant factor, right? I mean, I find that really, really surprising. Not wrong, I should emphasize, surprising and interesting. Like, I believe it, to be clear. I was curious, rather than about the transmission rate, how that range overlap might influence the persistence of some of these infections in groups. So do you find that kind of the persistence of it bouncing around the group might be longer if there's more overlap? What about the other ways in which we often measure disease and its, and it's how serious it is?
Yeah, we didn't directly test for that. I should say with these outbreaks, they were pretty short. So it was, some of them were kind of only a week, some of them were kind of 2 weeks in terms of like before everyone became infected. And then gradually, they got better, but the damage was done. I'm trying to think. Yeah, we didn't look for kind of the length in terms of the length of outbreaks and what kind of the effect of home range overlap could be. We do know that there's a relationship between home range overlap and stress. So groups that have kind of whose home range overlap with more other groups tend to have a higher kind of stress, and that could influence kind of how long individuals are sick for. So that's a really interesting idea and something we could look for.
Great, thank you. Eric, there's a question that's come through for you. There are actually two of them, so I'll do the same thing. I'll take advantage of asking the first question to follow up with the second one. The first one is from Adrienne Chitayat in the Q&A, and the question is, the results from adverse taste deterrents look really promising, but is this a feasible strategy for farmers who want to implement them on a wide scale? And how do you think does it affect the ability of farmers to sell potatoes afterwards? Oh, hold on a second, Eric. Let me see if I can, I think I can unmute the individuals. I think I forgot to unmute you there.
Can you hear me?
Now, perfect. Yes, thank you.
Yeah. What we did was we tried to see if the test salvation could be a potential solution to discourage monkey from grading on potatoes. So it was a preliminary time of being widely used. So it needs to be a well-structured study that can be confirmed that the test salvation can be applied at a wide scale. We were trying something small, but for it to be applied by farmers, need to be tested widely and to see if it is feasible.
Yeah, thank you. In fact, Gustavo, who's now embarrassed that I keep pointing at him, he had the question too about whether the potatoes were really bait, hoping that they would eat those and then leave, as opposed to the actual crop that would be sprayed with the chili oil instead. So it sounds like it's in the early phases of trying to figure out the best way to use it, but it's definitely promising.
Yeah, it is, because we found that at least the smell, those who picked up the potatoes didn't eat the potato, because we think the smell is repelling them from eating the potatoes, not eating them, because none of the monkeys that visit the experimental area ate the potatoes, only sniffing at the potatoes and then throwing them away. So it has to be tested again to see how we can use this as a way forward.
Excellent. Thank you. There's a question here for Eduardo, and I think Eduardo, yeah, you're still there. Eduardo, the question is it more energetically expensive to move in highly angled turning paths? Put another way, what are the questions? What are the costs and benefits of directed versus highly angled movements?
Oh, that's a great question, actually, and I haven't been considered it too much. We have a person, Gabriela Yazini, that is looking at specific energetic costs of movements, but related to the question, I believe that too much turning angles or not too much directionality, maybe not related to too much energetic cost, but backland tamarins, they usually cover all their territories, so that's why that's the reason they are highly directional. So I would say that I'm not for sure, but I would say that the high directionality context, usually in large fragments, are related to a huge expenditure of energy, because usually they are spending they are in higher velocity and with highly directional turning angles.
Perfect. Thank you very much. And so we have about 2 minutes left. So I think this will probably be the final question. And I just realized that I don't think Sophie is there. So we'll post that one in the question later for the section. And instead I'll ask this question here, which is a question from someone who knows very little about primates, for Jacqueline Ballantyne. And that is it, so you answered the first question, which was about whether they preferred the infested ones, or maybe it was just whether there were more of them out there, or the ratio of availability to consumption. But is it possible that there are, what are the negative consequences of consuming infested fruits? Isn't it actually possible that they prefer them because they're more nutritious? They get all this protein from the things that have infested them. So while it may seem gross to us, it's actually something that makes a lot of sense from the primate point of view, because they get lots of nitrogen that they wouldn't get from the fruit itself.
Yeah, that's exactly it. So the theory is that these fruits, their nutrition values might be increased. And in situations where, when we think of primates getting proteins and that being a limiting resource in a lot of instances, we think of insectivores as going out and collecting butterflies and grabbing insects in that way to collect that resource. But it's highly energetically costly to go out and try to catch insects. So these infested fruits might be a really reasonable way for them to get that protein. And then additionally, we were asking, is the fruit potentially more digestible, and is it easier to open if the infestation has already occurred? So hardness might decrease and stuff like that. So we showed that infested fruits are less dense. They're usually the larger fruits, so they'd be in trees where the primate would want to be, because the fruits are larger in the first place. And additionally, yes, the fruits are higher nutritional value for that limiting resource. So not in all nutritional aspects, but for protein and nitrogen, yeah, we can make that assumption. Does that kind of answer the question?
Perfectly, and with perfect timing, it's time for us to wrap. So I really appreciate it. If anybody out there in the audience would like to post any more questions, please go to the session page on Whova where you'll be able to post your questions and people can respond. You're welcome to, if you'd prefer to follow up the discussion later, start a discussion group as well. And again, thank you very much to our speakers. Really fantastic stuff. You captivated a 12-year-old, so you must have done a pretty great job. So I appreciate it, and we'll see you all at the next event. Reach out. Talk to you soon. Bye-bye.
Bye.